Hello, we're glad you've joined us for this webinar, Comprehensive Proteomics and Lipidomics Strategies to Advance Disparities Research in Alzheimer's Disease. I am Michelle Ashton of LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is presented by LabRoots and brought to you by Agilent. To learn more about our sponsor, please visit www.agilent.com. Let's get started. You can pose questions to the speaker during the presentation while refreshing your mind. To do so, simply type them into the drop-down box located on the far left of your screen labeled Ask a Question and click on the Send button. Questions will be answered after the presentation. If you experience technical problems seeing or hearing the presentation, just click on the Support tab found at the top right of the presentation window or report your problem by typing it into the Ask a Question box. This is an educational webinar and thus offers free continuing education credits. Please click on the Continuing Education Credits tab located at the top right of the presentation window and follow the process to obtain your credits. I now present today's speaker, Renã Robinson, the Associate Professor of Chemistry at Vanderbilt University. To learn more about our speaker, please click on the Speaker tab found at the top of your screen. Renã, you may now begin your presentation.
Hi, thank you for the opportunity to present today. The title of my talk is Comprehensive Proteomics and Lipidomics Strategies to Advance Disparities Research in Alzheimer's Disease. And so what I hope to do is give you an idea of some of the kinds of research that are taking place in our laboratory right now that are not fully complete but are still ongoing projects. So to get started, I thought it would be interesting to look at what the aging demographic looks like worldwide. And if you look at data from 2017, and what you see that on average across most countries, 1 in 8 people over the age of 60 in 2017. And when you look at the lower graph that shows you the distribution of what that looks like across different countries, then you can see that North America and a lot of Europe and Australia have a high population of older individuals. However, what's predicted to happen by 2050 is that 1 in 5 individuals will be over the age of 60. So this is the next 30 years, and you can see how this demographic would shift across the world and that there will be more individuals that are going to fall into this older demographic. And this becomes interesting to think about in the course of studying disease because there are a number of diseases where age is the biggest risk factor. And so here I'm showing you data from the U.S. from 2016 to 2017. And what you can see here is that in the United States, heart disease and cancer are the top 2 killers of individuals. However, there are a number of other different kinds of diseases such as stroke, diabetes, kidney disease, and Alzheimer's disease that also make it into this top 10 list of causes of death. And so for our group, we've been really interested in looking at Alzheimer's disease. And so you can see here that it's the 6th leading cause of death in the United States. And what we find interesting in terms of statistics about Alzheimer's disease is the fact that while diseases such as heart disease, the therapies and the treatments for these have decreased the number of deaths that have occurred over time, the number of deaths that occur from Alzheimer's disease have steadily been increasing over the time when you look at data over the last 15 years or so. And so currently there is no cure or effective treatment for Alzheimer's disease that can stop, delay, or slow the progression of disease. And so there's really a lot of work that needs to be done in order to reduce the number of people that are suffering from this particular disease. What we also find interesting about Alzheimer's disease within the United States is the fact that there are racial and ethnic disparities that exist when you look at the prevalence of disease. And so, for example, here I'm showing you data for the prevalence of Alzheimer's disease in African Americans compared to non-Hispanic whites, which is the purple trace. And what you see is that as aging increases, there's a larger gap between these 2 different ethnic groups in terms of prevalence of AD. So African Americans are 2 to 3 times more likely to have Alzheimer's disease than Caucasians or non-Hispanic white individuals. Hispanic individuals in the U.S. are 1.5 to 2 times more likely. And so you can see that even though African Americans make up a small percentage of the U.S. demographic, that they make up a large percentage of individuals that will be suffering from this disease moving forward. What's also interesting is the costs that are associated with Alzheimer's disease care and caregiving costs, and you can see that a small percentage of the population that is comprised by African Americans represents 1/3 of the total costs that are spent on Alzheimer's disease and other dementias. And so this presents a lot of socioeconomic and demographic issues in the United States. We're trying to figure out how to reduce these disparities for this and for other groups. So we've started to think about what some of the contributions are for this particular disparities. And so if you look at this chart, then what you see on the left is that there are different kinds of comorbidities. So things like heart disease that we mentioned prior, high cholesterol, hypertension, obesity, and stroke that contribute to Alzheimer's disease pathogenesis. So these are all risk factors for AD in addition to aging. And then there's certain genes such as ApoE4, which you've heard a lot about, and other genes like APCA7 and SERL1, which end up being potential genetic risk factors for African Americans with Alzheimer's disease. And so these different comorbidities and genetic factors can directly impact pathological hallmarks in Alzheimer's disease, including things like amyloid beta deposition and oxidative stress, and then ultimately to Alzheimer's disease. However, at this point, there's not a clear understanding of what the complete molecular basis is for these racial disparities that exist within Alzheimer's disease. When one looks at the relationship between what happens inside of the brain and outside of the brain in Alzheimer's disease, I wanted to bring to your attention a few things that we and others have observed. So this is coming from work by Trancon and al., who are demonstrating here in this chart that the brain isn't the only place where amyloid molecules exist, but also they potentially exist in the heart, and that these molecules can be trafficked between the brain and the heart. And so this can work both ways, where these deposits can impact amyloid beta in the brain and influence Alzheimer's disease pathology, but also having these amyloid deposits in the heart can impact heart function. And so this lends some idea to the thought of whether or not Alzheimer's disease is a systemic issue. And just to further hone on this point, these are data that come from that same study that essentially show you over here that the levels of amyloid beta 40 and 42, when you look at control individuals compared to those that have Alzheimer's disease, that those levels are higher in heart tissue, similar to how those levels are well documented to be higher in the brains of Alzheimer's disease patients. So this is one point to us that we find very interesting, that shows that other organs are also having a role in Alzheimer's disease. And then even in our own lab, we've done some studies recently with proteomics over the years, where we were able to demonstrate that in looking at liver tissue from an Alzheimer's disease mouse model, we observed higher levels of proteins involved in beta oxidation and ketone body formation in the liver. And we believe that these were showing us that elevated levels of ketone bodies were being generated in the liver and possibly trafficked to the brain to compensate for lower levels of glucose. And so these were further supported by some metabolomics studies that were done that demonstrated that, in fact, the levels of these small molecules were higher in the liver tissue. Okay, so I've given you a lot just in terms of background about how we think about Alzheimer's disease in our lab, and really all of that is to show that we really think that there is a role for other organs in the periphery in disease pathogenesis for Alzheimer's disease. And so our lab is really focused on trying to figure out what the role is for these different organs and how do these different systems contribute to racial and ethnic disparities within Alzheimer's disease. So what I'm going to do today is to walk you through 3 short vignettes about some of the work that's ongoing in our lab. And I'll start with a study where we've been looking at the relationship between the brain and periphery in mouse models and in human tissues of Alzheimer's disease. So one might envision that the goal for this particular project is to really understand the relationship between the brain and periphery. And so this just gives you an illustration of how we think about this. So if you have different organs that exist either in a mouse model or in the human tissue, and you think about a pathway such as metabolism, then you can see that different organs have different enzymes that might contribute to the overall metabolism that's taking place in the body, in addition to the lower levels of metabolism that are well documented in Alzheimer's disease. So our idea is to figure out if we start to look very early on prior to disease onset and at different stages in all of these different organs and in the brain, can we really start to figure out system-wide what are the initiators of these metabolic changes or other changes that are taking place in this Alzheimer's disease system. So for this work, we've used an Alzheimer's disease mouse model system. This is a system that is a human double transgenic knock-in mouse model. So it has human Swedish mutations in amyloid precursor protein and presenilin 1. And we've collected tissues from this mouse over the course of 3, 7, 12, and 14 months. And what you notice in this model is that as it gets older or disease progresses more, you go from an animal that is similar to a wild type at 3 months to an animal that starts to exhibit early stages of Alzheimer's disease with sparse amyloid deposits in the brain, that in later stages has numerous amyloid beta deposits similar to some of the pathology in humans. And so this isn't a fully comprehensive model of Alzheimer's disease, but it is one that allows us to get some insight about changes that are taking place in the disease. And the studies that I'm going to talk about, we looked at 14-month-old animals. So one of the projects that we have ongoing is done by graduate students, now postdoc in the group, Christina King. And she was interested to understand system-wide changes in Alzheimer's disease using a mouse model. And so what we did was we took wild type in our APPPS1, which I'll refer to as the AD mouse model. And in each of these models, we were able to harvest liver, brain, and heart tissue from each individual animal. And so we ended up with biological replicates of 6 for each group and a total of 36 samples. And so in order to run these samples using proteomics in a comprehensive way, we decided to employ a technique that we developed in the group that relies on quantitative proteomics. And so this is a technique that we developed years back, and it's called combined precursor isotopic labeling and isobaric tagging. And the way this strategy works is we take tissues that can come from wild type and AD animals. So over here on the left part of this graph, we have 3 pairs of wild type and diseased animals. We take the tissues from those, harvest the proteins, and generate tryptic peptides. And to these tryptic peptide mixtures, what we then do is lower the pH of the buffer, and we do a reductive dimethylation reaction, which adds the dimethyl group to the N-terminal amine of the peptides. What that does is it leaves the lysine amine open for further chemistry and tagging. So then we use these commercial tandem mass tags in order to label lysine residues. So each of these 6 individual samples gets one of the 6 different kinds of TMT tags that you can get that generate these reporter ions shown from 126 to 131 m/Z. And so we have 6 unique tags over here on the left. We do the same thing with a different set of tissues and animals over here on the right. And the only difference here is that when we do the dimethylation reaction, this dimethylation reaction consists of heavy isotopes. And so there's 2 carbon-13 and 6 deuterium. And so we add that tag to the N-terminal amines, and then we're able to use the other half of a TMT reagent vial to do the lysine tagging. So we're able to save cost here, but also expand the amount of sample multiplexing that we get. So all in all, we take 12 uniquely tagged samples, mix them together, and then we do one comprehensive multidimensional LC-MS experiment, where in this case we might use strong cation exchange coupled to reverse phase chromatography. An example of what you would see when you get the data is shown here, where in the full MS scan, we would see pairs of peaks that are separated by a mass of 8 Daltons for each dimethyl group that we've added, so our light and our heavy pairs. With our MS-MS data, we require selection of the light or the heavy peaks independently in order to get sequence coverage. And then we isolate the most intense, or with more advanced instrumentation, the top most intense ions, to then fragment again and generate MS3. So the most important part of this experiment is the MS3 spectra, which give us reporter ion data from each sample and inform us on the relative concentration of peptides and proteins and how they're changing across different conditions. Okay, so we employed this strategy to Christine a set of samples, and here's an example of the data that we got from a Fusion Lumos on the left. So here you can see an example peptide that has been dimethylated on the N-terminus and has been tagged with a TMT6-plex reagent on the lysine residue. And if you just look at C, then what you can appreciate is that within this set of data, we can see the difference in level of this peptide between a wild-type animal compared to a disease within a heart tissue, a brain, or a liver tissue. But then if you look across these 12 peaks, then we can actually start to see the differences across biological replicates and within tissues. So we think this experiment is pretty powerful. We've taken this data and we've ran the samples on different instrumental platforms as we had access to better technology. So we started on an Orbitrap Velos instrument, and you can see we identified about 2,200 proteins. We then moved to a Fusion Lumos instrument, and we did this experiment without any SCX fractionation, with incorporating SCX fractionation, and you can see with the Lumos we get about 5,000 proteins identified across the tissues. And then here you can see how these proteins overlap across the different platforms that we've used. What's most interesting to us about this kind of data is what happens when you start to tease apart specific kinds of pathways. So here's an example of what happens when you look at glycolysis system-wide. So here I'm showing you the glycolytic pathway, and these are the proteins and enzymes that we've observed in that pathway using this proteomics data set. And so, for example, if you look at glucose-6-phosphate isomerase, what you can see is that the level of this protein in the Alzheimer's disease brain tissue is lower compared to that in the wild-type brain. Similarly, the same is true for the heart tissue, where the levels are lower in the diseased animal compared to the wild type. However, in the liver, you see a different behavior, where the levels of this enzyme are higher in the diseased animal compared to the wild type. And so you can look across this slide at the different proteins in the pathway and see how they're changing. And what this does is it gives you a sense of the overall metabolism that's taking place in the entire animal, and not just within the brain tissue, where we mostly focus on disease pathogenesis. And interestingly, what you see is that there appear to be a lot of things that are happening in the liver tissue that are in concert with things happening in the brain and the heart that are most similar to each other. So we have lots of these kinds of stories when you look at different kinds of biological pathways. And to further support what we're seeing with the glycolytic pathway, there was a recent metabolomics study that came out by Zing and co-workers. And what this one shows is that if you look at the metabolites in this same AD mouse model across liver, kidney, and heart tissues, is that you start to see a lot of metabolites that are changing in energy metabolism pathways as well. And so what they identify in their work is that very early on in the liver tissue, you start to see changes in energy and metabolism that precede changes that take place in heart, liver, and other tissues. So we think this presents a really good way to start to tease apart what's happening in AD in early stages by focusing on tissues outside of the brain. But there are some caveats to using animal model systems to fully understand disease pathogenesis. And so while there's a lot that we can do with animal model systems, such as being able to harvest tissue from multiple organs, being able to use a smaller number of N in order to get good statistical power, being able to do this in a much faster time than we could do with human samples, and all of the tricks that we can play with genetics, there are the cons that sometimes the things that we find in these model systems aren't always directly translated into human studies. And that's because there's so much more diversity in human systems that aren't accounted for in the mouse model systems. And those could be things like socioeconomic, geographical, other kinds of diversity. And that the systems that we work with when we get data from humans aren't as well controlled as what they would be from a mouse model system. So that's led us to try to explore doing similar kinds of studies in human systems. And so here what we've done is we've done a pilot where we got some whole body tissues from a brain bank in Arizona, Bennett Institute. And we were able to get three sets of control and 3 sets of AD individuals where we could get the heart, brain, and frontal gyrus tissue and liver tissue from these individuals post-mortem. And so we did a similar quantitative proteomics study using our C-palette strategy on these sets of tissues. And we wanted to see how relevant were the things that we found in our mouse model to this set of human tissues. What this slide shows you is the overlap that we got in proteins that we identified across the different tissues. So here on the left, you can see in our AD mouse model, we see very substantial overlap in proteins across different tissues. When we look at the human data here on the right, then you can see that, again, we have very consistent overlap in proteins that we observe across the tissues, which pretty much means that we get coverage across most of our reported ion channels for each protein. But you also see that the number of proteins that we see is fairly similar between our mouse and our human system. This is a heat map that gives you an idea of the kinds of changes that we see overall across the different tissues. So here on the right is one example area where we zoomed in, and you can clearly see from the heat map that there are substantial changes that are taking place in the brain, indicated by the red colors, which are more pronounced than those happening in the heart and the liver. However, when you start to look at differences between the control and the diseased individuals, then you can actually see that there's a lot of things that are happening in all 3 of these different tissues. And so to give you a sense for how we look at the data, this is just pulling out 2 proteins, alpha-enolase and tubulin alpha chain, in which we wouldn't expect much activity with disease. And so here you can see that the levels are higher for both of these proteins in the brain tissue compared to the heart and liver, and there doesn't appear to be much change happening between the control and the diseased individuals. So we can keep mining the data in this way, but it's also easier to just kind of look at an overview of the data. And so these volcano plots give you a sense for the global changes that we see in each of the tissues. And so what we've done here is we've aligned the same tissue from the mouse model with the human brain tissue, heart to heart and liver to liver. And so you can see that there are some differences that might be observed when you look at the mouse compared to the human system. So for example, if you look over here at the liver, you can see that in the mouse model system, we see more proteins that are have higher levels in the diseased animal compared to the wild-type animal, whereas in the human system, we see more proteins that have lower levels in the diseased animal or in the diseased individual compared to the control individual. So we're working through the data now to figure out how much similarity or homology there are in proteins that are different across disease and within different tissues. And I can kind of give you a preview that already what we see is that there are things that, some things that overlap, but a lot of things that are unique to the mouse model system or the human system. So we definitely want to continue to do this work and tease out what's happening. But overall, what we think we've been able to demonstrate with these kinds of studies is that there's a lot that's happening in other tissues and other organs and disease. And these are worthwhile investigations because they can help us better understand the pathogenesis of Alzheimer's disease, but importantly, they also help us understand what the relationship might be with the risk factors for Alzheimer's disease, such as the comorbidities that I mentioned early on. So the second story that I wanted to tell you about is one where we've been working over the last couple of years to get some pilot data to really start to investigate racial disparities that exist within Alzheimer's disease in the U.S. So these are all preliminary work, pilot studies, and so I'll just give you a sense of what we're doing. So what we did was we worked with the Alzheimer's Disease Research Center at the University of Pittsburgh, and the goal was to perform a small study with tissues that it had available from African Americans and non-Hispanic whites to understand whether or not there were molecular differences in post-mortem brain tissue. And so what we did was we got as many tissues as we could from the African American group that were cognitively normal and that had AD, and this was diagnosis post-mortem. And then we matched to that our non-Hispanic white participants. So our N for this study are fairly small. So for most of the groups here, we have an N of 5. And what Caitlin did in the study was to look across different brain regions. So here we mostly focused on the hippocampus, which has early changes that take place in AD. We looked at the inferior parietal lobal portion of the brain, which also has some changes that happen later in progression. And then we looked at the globus pallidus, which is a region that doesn't have much pathology that occurs in AD, and we thought that would serve as a good negative control for this study. And all of the experiments were randomized and blinded to Caitlin when she began the work. So again, we use different MS platforms to do these studies, where we started on one platform and then got access to more state-of-the-art instrumentation. So we started this work with an elite MS, and these are just TMT-based experiments. And so we had less than 900 proteins that we identified with that platform. We then did the same MS3 experiment on a Fusion Lumos, and we got up to about 3,400 proteins. And then when we just look at doing MS-MS with the TMT on the Fusion, then we see about 5,200 proteins that we've identified. So the platform here definitely influences what we get from the data. And if we just focus on the portion of proteins we identified from the hippocampus and compare that to a comprehensive study that was reported by Ping et al., where they looked at 27 different brain regions, then what you see here is that we have an overlap of about 2,800 proteins that were previously identified. So that gave us some confidence that we were seeing things that have been seen before in AD brain. But we also have a lot of unique proteins that we observed in our study that weren't reported in the Ping et al. Study, and similar for them. And so we think that every time these experiments are ran, then we can learn more about what's happening in the brain. For some of the changes that we observed with the TMT approach in our data set, we went and did a verification with Western blot. So this shows you some examples for beta-actin peroxyredoxin, fatty acid binding protein, and CAM-K2 alpha, where the changes we saw by TMT were supported by Western data. And then if we start to look at the hippocampal region, then here we can see from these volcano plots how proteins are changing with disease, but importantly, how they're changing with the different racial backgrounds that we looked at of the participants. So on the top, you see data from the non-Hispanic white population, where you can see there's almost equal distribution of proteins that are higher in expression in Alzheimer's disease compared to cognitively normal individuals. However, if you look at the changes on the bottom that are demonstrated for the participants that self-identified as African Americans, you see that we see more emphasis on the left where there are proteins that are lower in expression in the Alzheimer's disease individuals compared to those that are cognitively normal. So this was the first clue to us that there was something really interesting happening here in the data set. And you can see that for a few proteins that we've labeled, there's some things that are similar. So for example, the glial fibrillary acidic protein is one that seems to be pretty consistent just within Alzheimer's disease. So the next thing that I wanted to show you is how we've taken the proteins that are different between our Alzheimer's disease and cognitively normal individuals, and we've looked at them by racial ethnic group and then also by brain region. So if you focus your attention just to what's happening in the hippocampal region, then you can see that we observe less proteins that are similar between the non-Hispanic white and African American groups, so that's the 99. Then we observe proteins that are unique to each racial group. So these are all proteins that are changing in the disease, changing with Alzheimer's disease, but are appearing to be more unique to a racial group. We then looked at different brain regions, so the inferior parietal lobule and the globus pallidus regions, and we see similar behavior where there's less numbers of proteins that are similar between the two racial groups than there are proteins that are unique to a racial group. And so, albeit that the N for this study is small, we do think this points to the fact that there may be a lot of changes that are taking place in Alzheimer's disease that we've not been aware of because of the lack of diversity in the populations that have been analyzed. So when we focus on just some example networks that we've observed in the hippocampus tissue, then here you can see that the pathways that are unique to each specific racial ethnic group, shown here by the string networks in red, are representing different pathways. So for our non-Hispanic white population, the most significant changes were observed in cell signaling and endocytosis-related pathways, whereas in the African American AD group, the most significant changes were observed in metabolism and immune response and inflammatory pathways. So this we think is, pretty exciting, and we're working diligently to try to get more in so that we can increase the statistics of our study. I mentioned very early on in the talk that we've looked at without lipid metabolism might be an important factor in disease pathogenesis. So within the same data set in the hippocampus, we started to look at changes in lipid metabolism proteins. So here you can see we see more proteins involved in lipid metabolic pathways when we use the Fusion Lumos compared to the elite. And then what you see with these different plots are examples of proteins that behave similar within a given pathway. So for example, if you look at B, what you find with the black trace in the middle is that on average, proteins in this pathway behave very similarly between the non-Hispanic white group compared to the African American group, where these lipid proteins are lower in Alzheimer's disease compared to cognitively normal individuals. However, this is different for the proteins that are shown in D, where on average these proteins are higher in expression than the Alzheimer's disease from non-Hispanic white individuals, however, aren't appearing to change in the African American group. So I think these kinds of data for us showed us that a lot of the changes that we may be seeing in Alzheimer's disease when we look at the different ethnic groups may not necessarily be really dramatic or drastic changes, that sometimes these changes are going to be more subtle, but they're still worthwhile to identify. We then looked in a different peripheral fluid. So we looked in plasma tissue that came from participants that meet the demographics that are shown here. And so we started by just randomly selecting 10 individuals from each one of these groups and getting the pilot going. And this is work by Mustafa Khan, who will be finishing in the next year in the group. And what he's done is he also did a TMT-based proteomics experiment. And you can see here how many proteins he identifies across each patch. So in total, we were able to look at 2452 plasma proteins and see how they changed across the different groups. This gives you a sense for the reproducibility across the different batches that he ran. So he was getting very similar reported ion intensity across them. And most surprising to us is that when we started to look at proteins that were different between the ethnic groups, that his data in plasma were very similar to what we were seeing in the post-mortem brain tissue, where there were few numbers of proteins that were similar between the non-Hispanic white and African American group compared to those that were unique to the different racial groups. And you can see from the different pathways that are represented here by those proteins that the differences in the pathways that are driving those unique observations between a given racial group. So we're really excited about this work. It's an ongoing project that we're working on, and so we hope to share more in the coming months by way of publication with these data sets. Okay, so our primary focus in the lab so far has been using proteomics techniques to understand what's happening in disease pathogenesis with different models and different individuals that come from different racial ethnic backgrounds. So I thought I'd finish by sharing a last and short vignette that has to do with the work that we're doing to develop lipidomics strategies to complement the work that we're doing with the proteomics for these kinds of applications. So for these studies, we've been working on establishing a lipidomics platform to study lipids from human plasma. And so right now we have worked out a sample preparation method that takes advantage of Bly-Dyer, where we get our extracted lipids. And then we can take these lipids after we spike in an internal standard, and we expose them to 3 different kinds of strategies. So the first strategy is an untargeted lipidomic strategy using a thermal Q-exactive instrument. The second strategy is one that uses multiple reaction monitoring on a SciEx Q-trap instrument that targets about 1,200 different plasma lipids that have been validated. And then the last platform is one that looks at single reaction monitoring on a TSQ quantum ultra instrument, where we just focused on lipids involved in the cholesterol pathway. And all of these are subject to different kinds of data analysis and biostatistical approaches. And this just gives you a sense for the chromatography instruments and mass spec instruments that are used for these studies. So we start by spiking our mixtures with the internal standard mix that comes from Avanti. This mix has a good distribution of different kinds of lipid classes and also good distribution of concentrations of these lipids that show up. And so these are deuterated forms of the lipids. And for each sample that we analyze, we do 2 injections, so one to get positive ionization mode data, and a second to get negative ionization mode data. And so these are data that I'm showing you for the untargeted platform on the QEHF. And you can see that we see a good distribution of lipids across the different classes. And so you can better see that when we look at these pie chart distributions of lipids across different lipid classes. And so mostly what you see is that in both positive and negative ionization mode, we have about half of the lipids that we see are glycerol phospholipids. And then we see a large percentage of lipids that are fatty acids and also glycerol lipids and sterol lipids that are also observed. So we do get some better performance of ionization for different lipids in the negative ionization mode than positive. So for example, phosphatidyl are better ionized in negative ionization mode than what we see here in the positive ionization mode data. So we took a small subset of lipid samples from Mustafa's plasma cohort, and we just had 5 cognitively normal individuals and 5 AD individuals, and we subjected them to reproducible experiments that we ran on 3 different days. And so these are not batch corrected so that you can see the differences between the batches. And so what you see from the PCA plots are that we can distinguish between the cognitively normal individuals from the diseased individuals very clearly. And then you can also see that there are differences between the days. And so those things are being normalized. And then what you see here on the right is that we see differences in the lipids that we observe between our Alzheimer's disease compared to our cognitively normal individuals. So we look at these by lipid class, and that's represented here on this bar chart, where you can go across the different classes that we observed in the untargeted analysis. And for most of the classes, we see changes that are consistent with what's reported in the literature. So for example, the first column is the phosphatidylcholines, and those are lower in expression in Alzheimer's disease, and so that's what we see is consistent with that. But then you see other pathways such as the lysophosphatidylethanolamines that are higher in expression in the Alzheimer's disease compared to the cognitively normal individuals. So we're working on, again, getting more in to beef up these data, but these just gave us a sense that we could begin to do these analyses with this untargeted platform. You can always mine the data further to go in and look at an individual lipid species of interest, and so here you can see many of those where you can start to tease apart how they're changing with the disease. So we worked a lot on the untargeted platform, and then we were able to establish a partnership with SciEx where we could look at targeted lipids. And so here with their platform, they have 1250 lipids that they know exist in plasma and that have been tested. And so here what you see on the top is an example trace using Hillock that shows you the separation of the different lipid classes that have all been labeled pretty clearly. And then what's impressive about what you see on the bottom is that we've taken our 30 different samples, so these were the different days of batches of samples that we had, and ran them after each other nonstop. And you can see just how reproducible the chromatography is for these particular species. You can go into any specific point in time here on the chromatogram, and then you can get back an annotated spectrum from the platform. So here you can see for the phosphatidylethanolamines, peaks that have been identified previously using this data. And so we can get that kind of information for each lipid class. And right now we're just really trying to learn how to handle these data and work through them, but we're really excited about the quality of the data that we're getting from both platforms. And even though I'm not showing you that, we do have some data that support we see similar changes with the targeted platform as we do with the untargeted platform. And then if you want to go in and further mine a specific pathway in lipid metabolism, here we're able to follow cholesterol and its precursors and a specific pathway using a very targeted SRM approach. And so here down the left, you can see the different cholesterol and precursor species that we've looked at, the transitions that have been identified by mass to charge, and then you can see how robust the chromatography is for identifying these particular species with this assay. So this is done in collaboration with a colleague here at Vanderbilt, Ned Porter. And for these studies, the only species in this pathway that we observed a difference for was cholesterol between the cognitively normal and the diseased individual. Okay, so thank you for hanging in there with me over the course of the last 45 minutes or so. I hope what I've been able to show you are a lot of the different kinds of projects that we're working on in the lab. I hope, importantly, we've been able to show you how enhanced multiplexing with proteomics is very useful for giving us the ability to study Alzheimer's disease using different tissues in the periphery and in the brain, looking across different disease stages in a way that we believe is very high throughput and cost effective. I also hope that we've shown you just how dynamic the changes are that we see in energy and lipid metabolism pathways as well as others when you start looking at heart and liver alongside the brain. And this was true for both our 80 mouse model and the human tissues that we observed. I showed you some interesting data that is very preliminary, looking at racial ethnic differences in AD, and what we observe in postmortem brain and in plasma are that there are differences that exist. And so we're still trying to get more statistics to better understand what these differences are, but we think they're important to helping us think about, one, understanding disease pathogenesis, but also beginning to think about how treatments should be established for Alzheimer's disease in personalized medical kinds of ways. And then I hope that I've shown you some of the fun that we've been having learning about lipidomics and trying to develop strategies there that we believe can help complement the work that we're doing with the proteomics. All of the work that we've done and that I've talked about was all made possible with mass spectrometry and different kinds of mass spec platforms. I'd like to thank my team who's contributed significantly to the work that we've done, our collaborators at the University of Pittsburgh and also here at Vanderbilt that have helped to support the work that we've done with lipidomics development. And then I'd like to thank our funding agencies, Banner Health for helping with the providing samples for the human tissue work. I'd like to thank Agilent for sponsoring this session. And again, thank you for your attention, and I'm happy to answer any questions.
Thank you, Renee, for your presentation. A quick reminder for our audience on how to submit questions. Simply type them into the drop-down box located on the far left of the presentation window labeled Ask a Question and click on the Send button. Renee will answer as many questions as time permits. Okay, let's get started. Our first question is, why do you think the overlap in protein identifications from the various MS platforms you used for the mouse work was low compared to all of the proteins identified?
Yeah, that's a good question. So what we've learned every time that we run these experiments is that we don't always see the same things, albeit we do see a good number of the same things. So I think a lot of it has to do with some subtle differences in the way that we carried out the fractionation as we evolved over the years from the VLOS platform to the MS. I also think a lot has to do with the sampling of how the data are sampled in the mass spec from the VLOS to the fusion. So what's very critical about these experiments is that we really have to observe and isolate both our light and our heavy precursors in order to get back robust reporter ion data. And so in that, I think if only a single pair was a single peak of the pair was selected in the VLOS platform or in the fusion, then we wouldn't have as much overlap between the techniques. And so certainly we've observed that with the LUMOS, we're able to increase our sampling of light and heavy pairs, and so we get more comprehensive data with that platform.
Thank you. Do you see all lipid classes with both the untargeted and targeted MS approaches?
So no, so we don't see all lipid classes. I do think we see a majority of the lipid classes that are observed. I think the platform itself, what most influences our ability to see the lipid classes are the front end separations that are used. So in the way that we've been working on this, we ended up switching from a reverse phase separation that we were using for the untargeted to a helic-based separation for the targeted. And we found that the helic gave us more robust coverage of the different kinds of lipids, but there were a couple of classes that we still didn't observe with the helic separation either in the targeted platform. So I think, if there are specific classes that you want to go after, definitely doing a more targeted separation approach to get them is going to be necessary.
Now, how critical is the MS platform to the quality of the data presented for the proteomics work?
Oh, it's very critical. Hopefully I was forthcoming in the data that we showed throughout, just showing the evolution of the quality of the data that we got from Velos and Elite platforms compared to Fusion Lumos platforms. I think the mass spectrometer plays a big role because the performance of the mass spec by way of sensitivity and duty cycle and how fast you can generate different scans is very critical to being able to get as much data as you can, but also as much data of good quality. So certainly we've observed, with better instruments in time, we've been able to get better quality data. I will say that I don't think that you necessarily have to have some of the best instruments to get quality data. I think it just requires more work on the front end and in the way that you do your data collection in order to get comparable amount of information. But yeah, hopefully that answers the question. Definitely with better quality instruments, we get better data overall, whether that was with our proteomics workflow or with our lipidomics workflow as well.
Thank you. Okay, it looks like we have time for one more question. Have you considered ways to increase the sample size for ethnic groups, and also to look across other racial ethnic groups?
Yeah, so we certainly were naive when we went into this project and thought it would be very easy to get the large sample sizes for different ethnic groups, and we realized that that would require more work. But we've been working diligently to establish collaborations with groups in the U.S. that have cohorts where they look at African American groups so that we can increase our sample in there. And we've been interested to also look at other racial groups. I think that will come with time once we can build up these collaborations to have access to samples, and also once we can demonstrate that we have a good handle on what is necessary to get good quality data that's going to be powered well statistically, but also start to learn what the differences are. But I definitely think in the future we have to look across different ethnic groups, and I would say racial and ethnic groups, so that we can have a good understanding of what's happening with disease. Because, for example, Japanese Americans have an extremely lower prevalence of Alzheimer's disease compared to other groups. And so I think, that's interesting to follow up and study as well.
I would like to once again thank Renee for her presentation. I'd like to thank the audience for joining us today and for their interesting questions. Questions we did not have time for today will be addressed by the speaker via the contact information you provided at the time of registration. We would like to thank LabRoots and our sponsor Agilent for underwriting today's educational webcast. LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Until next time, goodbye.